Good morning, everyone. Today, I would like to present my work entitled Rejuvenation of Two Models of Aging Rats by Introducing DNA Gaps. This figure, our PI, Professor Apiwat Puthirangun, already introduced you the discovery of DNA protection and rejuvenation effects of DNA gap by this molecule, the molecular scissors molecule. And this session, I would like to introduce you the implication of these molecular scissors in animal model. Here, let me start first with the two model of aging. The first one, we use D-galactose-induced aging rats, and another one, we use the naturally aging rats model. And we detected the number of DNA gap, and we found that during aging, the significant decrease in the number of DNA gap found in both aging model, as shown here. And then in our study, we treated animal with D-galactose. We treated an animal with the molecular scissor molecule by weekly injection of the plasmid expressing this molecule for 8 weeks. And then at the end of the study, we detected the DNA gap number. After treatment, we found a restoration of DNA gap number in both models. However, we also study the mutant of these molecules that lost the function of the molecular scissor and cannot produce the novel DNA gap into the DNA. And we also detected the liver function tests, including the serum, the rat serum, AST or aspartate aminotransferase, serum ALT, alanine aminotransferase, and alkali phosphatase as well. The results show that the significant decrease in the liver function enzyme show in both models, decal aging and natural aging. However, the total protein, serum albumin, and globulin show no significant differences among all groups. Moreover, we also measure the visceral fat weight after treatment. And we found that after 8 weeks of treatment, the decrease in the visceral fat weight found in both models, but not in the mutant. We also explore the histopathological appearance in rat liver, and we found the significant improvement in the appearance after treatment in both models. And we also measure the sinusoidal space in liver section. We measure by using the ImageJ program, and we found a significant decrease in sinusoidal space after treatment in decal aging rat, and we found a trend of the decrease in the aging model, but not significant. In addition, we also measure the collagen decomposition or collagen accumulation by mesocycle method. And the blue stain here show the deposit of the collagen. And we found that after we analyze with the ImageJ program, we found a significant reduction of the liver fibrosis in both aging models. Moreover, we also measure the aging marker. First one, we detected the senescent-associated beta-galactosidase or SA-beta-gal. And after treatment, the D-gal aging rat showed improvement of the aging marker. Similarity, it is similar to this model. After we treated with the molecular sister molecule in natural aging rat, we found the improvement of aging marker as the beta-gal staining as well. After that, we also quantitate the the percentage of SA beta-gal positive cell, and we found a significant reduction of these aging marker in both models. Moreover, we also extract the protein from rat liver and use the Western blot assay to detect the aging marker P16, P21, as well as the DNA damage marker gamma H2AX and the upstream signaling P53. And we found the significant decrease in the DNA damage marker here in both model, but not in mutant, as well as the aging marker late state of senescent P16 and P21 also reduced in both models. But this data show no significantly difference in P53. And here we also study the learning and memory brain function of the brain or the cognitive function by using the Morris-Waterman test. We found that in both models showed improve in learning function of the brain on day 4 and day 3 and day 4 in both models. And surprisingly, we also observed the improvement of memory on day 6 in both models, but not by treatment with the mutant. And we also test the memory using another method by novel object learning, and the similar result show here. We found a significant improvement in memory in aging rat. So here let me summarize what a new finding in our study. The study of molecular sister molecule intervention. We use two models of aging, D-galactose-induced and natural aging rat model. We found the improvement of learning and memory in the brain. And this treatment can restore the number of DNA gap in white blood cell DNA of aging rat. Importantly, in the liver, we found the improvement of liver function parameters, and we found the reduction of DNA damage gamma H2AX and aging marker SA beta-gal P16 and P21. Moreover, we also observed the improvement of anatomical appearances of aging liver tissue, as well as we show the reversal of liver fibrosis in aging rats. Surprisingly, we see the significant decrease in visceral fat weight in aging rat as well. So let me thank to our PI, Professor Aphivat Muchirangul, from that provide us the funding and the insight of this study. from Center of Excellence in Molecular Genetics of Cancer and Human Disease, Faculty of Medicine, Chulalongkorn University, and also AM Lab member. And moreover, I would like to thank Professor Nipon and Professor Siriporn Satipakon from Chiang Mai University that they are our collaborators to test the novel object learning in rat model. And as well as I would like to thank Dr. Suamsida Supasai for and her team for testing the brain protein expression in rat model as well.